today we are discussing about formation of germ layers. So we know that we have three germ layers: ectoderm, endoderm and mesoderm. So let us know how the germ layers are formed. First, we know that the zygote has two cells. Yes, the zygote has two cells. So the zygote is formed by the formed at a fertilization by the union of male gamete as well as female gamete. So the zygote consists of two cells. Out of these two cells, the large cell divides first into two. The large cell divides first into two by cleavage division, and then the other cell, it also divides. Okay, sorry. So the large cell divides first and it divides into two cells by cleavage division, and again what happens is the third cell again divides into two. So the cells, they keep on dividing, the cleavage division continues, and here the cells reach a 16 cell stage. The cells keep on dividing at 16 cell stage. The cells, that means this is a zona pellucida, right? So at the 16 cell stage, we called this group of cells, we call them as morula. So at the 16 cell stage, we call them as morula. Okay, so first the zygote consists of two cells, and these two cells undergoes cleavage division, and at the 16 cell stage, we called these cells as morula. Now what happens here is these cells, they are arranged into two masses. When we take the section of the blastocyst, we can see here the cells are arranged in two layers. The outer cells here, they are arranged surrounding the zona pellucida, at the periphery of the zona pellucida, and the inner mast cells. So outer mast cells and inner mast cells, the cells are arranged in two masses. Okay, so outer mast cells and the inner mast cells. So this is the zona pellucida. So what is the function of zona pellucida? So this zona pellucida, these are the trophoblast cells and these are the embryoblast cells. Okay, so see here, these are the trophoblast cells, the cells lining the periphery, of the zona pellucida. These are the trophoblast cells, and the inner mast cells are called embryoblast cells. So these are the trophoblast cells. Okay, trophoblast cells, and these are the embryoblast cells. So this is the zona pellucida. Okay, so what is the function of zona pellucida? We know that the fertilization takes place in the amble of the uterine tube. So because these cells are formed after fertilization, and zona pellucida helps in the formation of the embryo, which helps in the proper site of implantation, because this trophoblast has the capacity of eating the other cells. So because of this trophoblast, if the zona pellucida disappears, it results in the abnormal site of implantation, because this trophoblast has eating the other cells and it implants immediately. So this zona pellucida, it helps in the proper implantation at the upper segment of the uterus. It helps in the proper implantation in the upper segment of the uterus. So as the cells are forming, inner cell mass, outer trophoblast cells and inner cell mass, embryoblast cells are forming, the zygote which is formed, the blastocyst which is formed, it comes to the upper segment of the uterus. Upper segment of the uterus means consists of fundus and upper part of the body of the uterus. It constitutes the upper segment of the uterus. Now what happens here is, after reaching the posterior wall of the body of the uterus and fundus of the uterus, what happens here is the trophoblast gets implanted into the upper segment of the uterus between the fundus and the body of the uterus. Okay, so here the trophoblast gets implanted into the posterior wall of the uterus in the upper segment of the uterus. Then only the zona pellucida disappears. Then the trophoblast, it enters, it gets implanted into the uterine lumen. See here. So first of all, two cell stage, zygote consists of two cell stage. Then what happens is undergoes cleavage divisions and forms a blastocyst this blastocyst at 16th cell stage we call it as morula. And then what happens here is the cells they arrange in outer cell mass and inner cell mass. Outer cell mass cells we call it as trophoblast, and inner cell mass cells we call it as embryoblast. And this zona pellucida prevents the abnormal site of implantation, because the trophoblast cells have the capacity to lead the other cells and they get implanted anywhere. So the zona pellucida it disappears after the trophoblast, that means the blastocyst, after reaching the normal site of implantation, that is the upper segment of the uterus, that is the posterior wall of the fundus, junction between the posterior wall of the fundus and body of the uterus. Then only the trophoblast disappears. Then what happens? The trophoblastic cells here, they erode the uterine lumen, and the embryoblast, that is that the embryo, that means the blastocyst get implanted in the normal site. So this is the trophoblast, and this is the embryoblast. And the cavity here between the inner cell mass and outer cell mass, we call the cavity as blastocele. We call the cavity as the blastocele. This blastocele consists of fluid which is derived from the uterine lumen. Okay, so then what happens here is here the outer cell mass is the trophoblast and the inner cell mass is the embryoblast. Now what happens here is some cells of the inner cell mass. So this is the inner cell mass, right? This is the inner cell mass, nothing but the embryoblast. And this is the trophoblast. Some cells of the inner cell mass, they become flattened. There are some cells of the inner cell mass, they become flattened, which have represented with blue color. This is called endoderm, which is the first germ layer to be formed. So some cells of the inner cell mass, they differentiate and become flattened, and they form the endoderm. Now what have, so here is the, this is the, I have represented with black line is the trophoblast, nothing but I have represented the black line is the trophoblast. And now what happens is, next what happens is the remaining cells of the inner cell mass, they become columnar. The remaining cells of the inner cell mass, they become columnar and they form ectoderm. And this is the endoderm. Okay. And this is the endoderm. Now what happens here is, so this is the ectoderm, which is lined by columnar cells, and this is the endoderm, which lines the inside the trophoblast. We know that the black one is the trophoblast, right? This is the trophoblast. This is the ectoderm. Now what happens here is the endoderm comes to line the inside part of inside the blastocyst, inside the trophoblast. So the endoderm comes here to line the trophoblast, and this sac we call it as primary yolk sac. This sac we call it as primary yolk sac. Okay, now the black one we have represented is the trophoblast, and here we have ectoderm, and here we have the endodermal cells lines inside the trophoblast. And here what happens here is the ectoderm gets separated from the trophoblast. So the black one is called trophoblast, black line. So the ectoderm, which are represented with columnar red color, gets separated from the trophoblast. So, okay, see here that the ectoderm gets separated from the trophoblast. The area above the ectoderm, we call it as amniotic cavity. Amniotic cavity, which is roofed by amniogenic cells, amniogenic cells which are derived from the trophoblast here. So this is the amniotic cavity, and this amniotic cavity is roofed by amniogenic cells which are derived from the trophoblast, and the area which have represented with red columnar cells is the ectoderm, and here the endoderm lies inside the trophoblast, and now the blastocyst we call it as primary yolk sac. Okay, then what happens here is the trophoblastic cells, they multiply and they form extra embryonic mesoderm. This is called extra embryonic mesoderm, means it doesn't form inside the embryonic disc, it is forming outside this is the embryonic disc, and it is the trophoblast undergoes division and they forms extra embryonic mesoderm, which have represented with black color, that is nothing but extra embryonic mesoderm. extra embryonic mesoderm. Now what happens here is, see here, so this is the amniotic cavity, and the roof of the amniotic cavity consists of amniogenic cells which are derived from the trophoblast. Right. Now what happens here is, the cells, the flattened cells of the endodermal cells, they become cubical. The flattened cells, they become cubical. And what happens here is, there exist some spaces in the extra embryonic mesoderm. Some spaces are formed in the extra embryonic mesoderm. And then what happens is, these spaces coalesce to form a coelom, that is called extra embryonic coelom. This is the extra embryonic coelom, which divides the extra embryonic mesoderm into plankton-pluric layer of extra embryonic mesoderm, or visceral layer of extra embryonic mesoderm, and parietal layer of extra embryonic mesoderm, parietal layer of extra embryonic mesoderm. At one stage where the trophoblast, where the extra embryonic mesoderm is attached to the amniotic cavity, at one place this extra embryonic mesoderm does not extend, and in future it forms the connecting stalk, Now what happens here is, so we know that the endodermal cells, they become cuboidal, and the endodermal cells, the flattened endodermal cells, they become cuboidal and they line inside the yolk sac, and now this yolk sac we call this as secondary yolk sac. And now the extra embryonic mesoderm, the embryonic disc, it is suspended from the extra embryonic mesoderm. It is suspended from the extra embryonic mesoderm into the extra embryonic coelom. Okay, so this is how the secondary yolk sac is formed. So again, coming to the first, the zygote consists of two cells, and these two cells, they undergo repeated cleavage divisions and form a 16 cell stage. We call it as morula. 16 cell stage, we call it as morula. And again, these cells, they differentiate into inner cell mass called embryoblast and outer cell mass called trophoblast. Okay, then these trophoblast is covered by zona pellucida, which prevents the trophoblast for the abdominal sides of the implantation. It prevents the trophoblast to implant at the abnormal sites. So the zona pellucida, after reaching the uterine lumen, and it disappears by the action of trypsin-like enzymes, then this trophoblast, it is eroded the tissues of the uterine cavity at the proper site of implantation, that is the junction between the posterior wall of the fundus and the body of the uterus, that is the normal site of the implantation. This trophoblast get erode the tissues of the uterine level and gets implanted over there. See here. Now what happens here is now what happens here is, some of the inner cell mass, they become differentiated and become flattened cells. These cells forms the endoderm. That is the first layer to be formed. And next what happens here is, the cells present above the endodermal cells, inner cell mass cells, which are present above the endodermal cells, they become columnar. That is the second layer is formed, that is called ectoderm. Now what happens here is, these endodermal cells, they lie inside the trophoblast. This is called primary blast. Now this cavity we call it as primary yolk sac, and the embryonic disc we call it as bilaminar disc, consists of two germ layers, ectoderm and endoderm. Okay, then what happens here is, the trophoblastic cells, they proliferate and they form the extra embryonic mesoderm. Later on what happens is, some cavities appear in the extra embryonic mesoderm, and they coalesce to form a coelom called extra embryonic coelom, which divides the extra embryonic mesoderm into parietal layer of extra embryonic mesoderm and visceral layer of extra embryonic mesoderm. The mesoderm which is in contact with the embryonic disc, we call it as visceral layer of extra embryonic mesoderm, and the mesoderm which is in contact with the trophoblast, we call it as parietal layer of extra embryonic mesoderm. This extra embryonic coelom does not extend into the wall at the near the amniotic cavity, and in future it forms the connecting stalk. Okay, now here what happens here is, the flattened endodermal cells, they become cuboidal and they line the inside the amniotic, and they line the inside the yolk sac. Now this yolk sac we call it as secondary yolk sac, and this is the amniotic cavity. the amniotic cavity along overlying the extra embryonic mesoderm, we call it as amnion. Two important membranes are formed here. One is amnion and the chorion. So the one is amnion, it is consists of amniogenic cells and amniotic cavity excluding the ectoderm, we call it as amnion. So amnion is consists of the amniogenic cells forming the roof of the amniotic cavity. This is the amniotic cavity, right? The roof of the amniotic cavity is formed by the amniogenic cells and the floor is formed by the ectodermal cells. So amnion consists the amniogenic cells covering the roof of the amniotic cavity that forms the amnion. And what is the chorion? Chorion is the somatopleuric layer of the extraembryonic mesoderm along with the trophoblast. This forms the chorion. So chorion is nothing but the parietal layer of extraembryonic mesoderm along with the trophoblast, we call it as chorion, and the wall of the amniotic cavity constitutes the amniogenic cells excluding the ectodermal floor, that is forms the amnion. Amnion and chorion, they plays an important role in future developments. So that's about the two germ layers are formed, ectoderm and endoderm. And what about mesoderm? We will discuss them now. So, now how intraembryonic mesoderm is formed? We know that the extraembryonic mesoderm is formed by the proliferation of the trophoblastic cells. It lines the outside the embryonic disc and it doesn't give rise to any tissues or organs of the body. Okay, so how the intraembryonic mesoderm is formed? We know that the ectoderm cells are columnar, right? The ectodermal cells are columnar and the endodermal cells are cuboidal. And the endodermal cells are cuboidal. Here we can't define what is the head end of the embryo and what is the tail end of the embryo. Now what happens here is at one area the cuboidal endodermal cells, this is the ectodermal cells, right? Ectodermal cells which are columnar. And these are the endodermal cells, right? These are the endodermal cells which are cuboidal. Next, what happens? Cuboidal. So what happens here is at one side the endodermal cells, the cuboidal endodermal cells, they become columnar. At one side the cuboidal endodermal cells, they become columnar, and this columnar cells, this area we call it as procaudal plate. This area we call it as procaudal plate. So the cuboidal endodermal cells, at one stage they become columnar, and this plate we call it as procaudal plate. So this is the area where the procaudal plate is formed, that is the head end of the embryo, and this is the tail end of the embryo. Now we can define the axis of the embryo, which is head end and which is tail end, by the formation of procaudal plate. What is procaudal plate? It is nothing but the endodermal cells, they become columnar at one area. We call it as procaudal plate. Okay, so when we take the axis, the area where the procaudal plate is called, this is the head end of the embryo and this is the tail end of the embryo. The Y represents the tail end of the embryo, the X represents the head end of the embryo. This is the procaudal plate. Okay, now what happens here is, we know that the ectodermal cells are columnar cells, right? The ectodermal cells are columnar cells. Here we have procaudal plate is formed, with the cells become columnar here. Now what happens here is the ectodermal cells, we know that this is the head end of embryo and this is the tail end of embryo, right? The procaudal plate, where the procaudal plate is formed, this represents the head end of the embryo and this is the tail end of the embryo. Now what happens here is the ectodermal cells, they get proliferate near the tail end of the embryo. The ectodermal cells, they get proliferate at the tail end of the embryo and forms an elevation called primitive streak. The ectodermal cells, they proliferate at the tail end of the embryo and forms an elevation called primitive streak. Primitive streak. Primitive streak. So here primitive streak is formed. Here the primitive streak is formed near the tail end of the embryo by the proliferation of the ectodermal cells. By the proliferation of the ectodermal cells. Now what happens here is, from this primitive streak, the cells of the primitive streak, they pass in between the ectoderm and endoderm. So the cells of the primitive streak, these are the procaudal plate and these are the cuboidal cells, right? So the cells of this primitive streak near the tail end of the embryo, the cells of the primitive streak near the tail end of the embryo, they proliferate and they pass in between the ectoderm and endoderm. They proliferate and pass in between the ectoderm and endoderm. See here, these are the ectodermal cells and these are the endodermal cells. These are the endodermal cells. And these are the primitive streak cells which are derived from the ectoderm and they pass in between the ectoderm and endoderm and they form the intraembryonic mesoderm Now the germ disc consists of all the three germ layers. Now we call this germ disc as trilaminar germ disc. Okay, intraembryonic mesoderm through which most of the tissues are formed. But here one important point is, so from the primitive streak, the cells, they grow sidewards and they form the intraembryonic mesoderm. And here at the region of procaudal plate, where the ectoderm and endoderm are attached here. There the ectoderm and the endoderm, this is the procaudal plate, right? The cells, they doesn't extend into the procaudal plate. So this is the procaudal plate. The cells from the primitive streak, they doesn't extend between the ectoderm and endoderm. This forms the procaudal plate and it consists of only two layers, ectoderm and endoderm, and the mesoderm does not intervene, does not enter. In future it forms, this procaudal plate consists of only an ectoderm and endoderm. In future it forms the buccopharyngeal membrane. Okay, now what happens here is, so from the primitive streak, the intraembryonic mesoderm spreads from the sidewards. Okay, and also the intraembryonic mesoderm, it also spreads downwards, and it leaves a space behind the primitive streak, and this also, here also the ectoderm and endoderm, they are in contact with each other. Here also the ectoderm and endoderm, they are in contact with each other, which forms the cloacal membrane. Here also mesoderm does not intervene between the ectoderm and endoderm, and this is the connecting stalk, where the extraembryonic mesoderm is connected to the trophoblast. Okay, so here how primitive streak cells are formed? It is the primitive streak cells are formed by the proliferation of the ectodermal cells, and these primitive streak cells, they grow sideways and forms the intraembryonic mesoderm. So here the ectodermal cells proliferate and form a elevation that is called primitive streak. From the primitive streak, the cells extend sidewards, and here they form intraembryonic mesoderm, and they also from the primitive streaks, the entire embryonic mesodermal cells, they also extend downwards, but leaving a space behind the primitive streak, and where the ectoderm and endoderm, they are in contact with each other, it forms the cloacal membrane. So the precaudal plate forms in future forms the buccopharyngeal membrane, and here this forms the cloacal membrane. Okay, now we can see as the embryonic disc growing upwards, the primitive streak at present it is oval in structure. The primitive streak appears to be oval, right? The primitive streak appears to be oval. Now what happens here is, as the embryonic grows, what happens here is the primitive streak becomes a linear structure. From the oval structure to the primitive streak becomes a linear structure. So this is the procaudal plate. Okay, this is the procaudal plate which gives rise to buccopharyngeal membrane, and here the primitive streak becomes a linear structure. The cranial part of the primitive streak shows a thickening called primitive knot. The cranial part of the primitive streak shows a thickening. We call it as primitive knot. This one we call it as primitive knot. And here a depression present. At the center of the primitive streak, we call it as blastopore. Now we call it as blastopore. Now what happens here is, from the cranial end of the primitive streaks, the cells extends upwards up to the caudal end of the procaudal plate. The cells extends up to the caudal end of the procaudal plate to form a process called notocaudal process. So the primitive streak at present it is a linear structure. Then what happens here is, due to the growth of the embryo, it becomes a linear structure. This is the primitive streak, right? Then what happens here is, the cranial end of the primitive streak shows thickening. We call it as primitive knot, and in the center we have a depression that is called blastopore. Now what happens here is, the cells of the primitive streak, they proliferate from the cranial end of the primitive streak to the caudal end of the procaudal plate. They pass in between the ectoderm and endoderm, and they form a process called notocaudal process. So the notocaudal process, here this is the procaudal plate, and this is the primitive streak, this is primitive knot, and here is the notocaudal process is formed. So see here, so the primitive streak, from the primitive streak, the cells extend up to the caudal end of the procaudal plate. The cells extend up to the caudal end of the procaudal plate, and they form a process called notocaudal process. So it passes in between the ectoderm and endoderm. These cells pass in between the ectoderm and endoderm. So the notocaudal process and undergoes a series of changes and forms a definite notochord. Undergoes a series of changes and finally forms a definite notochord. So how definite notochord is formed? First, here we know that how the definite notochord is formed. We know that this is the ectoderm layer, and this is the endoderm layer. Okay, this is the procaudal plate. We know that the some of the endodermal cells, they become columnar, and this is the procaudal plate. Here notocaudal process is formed. By the proliferation of the cells of the primitive streak, notocaudal cells are formed in between the ectoderm and endoderm. Okay, then what happens here is, from the blastopore, we know that the primitive knot, at the primitive knot, we know that the primitive streak cells, they undergo proliferation and forms a thickening called primitive knot. And in the primitive knot, we have a depression called blastopore. So then what happens here is, so these are the ectodermal cells. Okay, and these are the endodermal cells. These are the endodermal cells. The blastopore, it enters into the notochordal process and forms a canal called notochordal canal. And forms a canal called notochordal canal. So this is the notochordal canal. This is called notochordal canal. Okay, then what happens here is the floor of the notochordal canal comes to break down. The floor of the, this is the ectoderm made up of columnar cells, and this is the endoderm made up of cuboidal cells. Okay, this is the endoderm made up of cuboidal cells. The floor of the notochordal canal comes to break down, and the cells of the notochordal canal, they come to lie along with the endodermal cells. They come to lie along with the endodermal cells. They come to lie along with the endodermal cells. So they come to, so these are the endodermal cells, right? So here we have endodermal cells are present, and these, the floor of the notochordal canal comes to break down, and the cells, they come to lie along with the endodermal cells. Now what happens here is the notochordal canal, the notochordal process comes in direct contact with the secondary yolk sac. We know that this is the secondary yolk sac, right? This is the secondary yolk sac, and this is the amniotic cavity, right? They comes into contact with the secondary yolk sac. They come into contact with the secondary yolk sac. Okay, now what happens here is again the cells, this is the ectodermal cells, and these are the endodermal cells along with some notochordal cells. Again what happens here is the notochordal cell, again they raised, and again they cut off from the endoderm and forms a definitive process called definitive process, that is nothing but the notochord. Definitive notochord is formed. That is nothing but the definitive notochord is formed. Okay, so how notochord is formed? Formation of notochord. Listen carefully. So here the primitive streak. We know that what is primitive streak? The endodermal cells. What is primitive streak? These are the ectodermal cells, and these are the endodermal cells, right? The endodermal cells at one place, they become columnar, and this is called primitive. This is columnar, and they form the procaudal plate. This forms the procaudal plate, and here the ectodermal cells, they proliferate and then forms here primitive streak. The ectodermal cells proliferate, they forms the primitive streak at the tail end of the embryo. The procaudal plates forms at the head end of the embryo. So the primitive streak, at first it becomes a oval structure, and later on due to the growth of the embryonic disc, it becomes a linear structure. The cranial end of the primitive streak shows a thickening called primitive knot. Shows a thickening called primitive knot. A depression is present in the primitive knot, we call it blastopore. Now the cells of the primitive streak, they pass in between the ectoderm and endoderm. The cells of the primitive streak, they pass in between the ectoderm and endoderm and form and extends up to the caudal end of the procaudal plate. This is the procaudal plate, the caudal end of the procaudal plate, and they form notochordal process. This notochordal process undergoes differential changes and forms the definitive notochord. What are the differential changes? We know that this is the notochordal process which is present between the ectoderm as well as this, which is present between the ectoderm, and below the cells are the endoderm, and this is the yolk sac. Below the endoderm, what we have? We have yolk sac is present below the endoderm, right? Because yolk sac is lined by the endoderm. Now what happens here is from the blastopore, the cells a canal extends into the notochordal process and forms the notochordal canal. And what happens here is the cells of the floor of the notochordal canal, they break down and come into lie with the endodermal cells. That means the notochordal canal, now it comes into contact with the yolk sac. Now what happens here is again the cells grows upwards, a rhizoid ridge is formed, and again they separate from the endodermal cells and they form definitive notochord. And here the notochord does not give rise to any important structures of the body, but at the region of notochord, vertebral column and ribs are formed at the region of notochord, but the notochord doesn't form vertebral column and ribs. Only the area which is occupied by the notochord now, it gives rise to vertebral column and ribs, but the notochord doesn't give vertebral column, does not give the development of vertebral column and ribs. Only the remnant of the notochord forms the nucleus pulposus of the intervertebral discs. The nucleus pulposus of the intervertebral discs intervertebral disc. We know that the intervertebral discs, they are present in between the vertebra, right? The intervertebral discs, they have two parts, annulus fibrosus and nucleus pulposus. So the remnant of the notochord in future degenerates, and the remnant remains as the nucleus pulposus of the intervertebral discs. Okay, so the notochord does not give rise to any important structure. A remnant of the notochord forms the nucleus pulposus of the intervertebral discs. So here the notochordal process, this is the definite notochordal process. A canal extends from the blastopore. A canal extends, that is called notochordal canal. And the floor of the notochordal canal breaks down, and the cells of the notochord, they comes to lie along with the endodermal cells. From again the cells, they get separated from the endoderm, and their definitive notochord is formed. So that is the fate of notochord, and in future it forms the nucleus pulposus of intervertebral discs. So it is about the formation and fate of notochord. So that's about the notochord. So now we know that the intraembryonic mesoderm is developed from the primitive streak. So this is called primitive streak. How primitive streak is formed? It is formed by the proliferation of the ectodermal cells. So the primitive streak becomes a linear structure here, and here the cells from the primitive streak, they spread sideways, and here the intraembryonic mesoderm is formed. And here is the notochord. This one is called as the notochord. We know that the notochord extends from the cranial end of the primitive streak to the caudal end of the procaudal plate, which in future forms the buccopharyngeal membrane. Okay, now what happens here is, on the top of the notochord, what happens here is the ectodermal cells proliferate. On the top of the notochord, the ectodermal cells proliferate, and they form the thickening that on the top. So this is the notochord, and this is the mesoderm here. So the mesoderm here is the intraembryonic mesoderm. Because it lies within the yolk, within the embryonic discs, right? That is what we call as intraembryonic mesoderm. The ectodermal cells becomes thickening on the notochord and forms the ectoderm. This is the ectoderm and this is the endoderm. So this is the ectoderm, okay, and this is the endoderm. Now what happens here is the ectodermal cells, they proliferate on the top of the notochord. This is called neural plate. This is called neural plate. Then what happens here is the neural plate, it undergoes depression, it gets depressed, and here the neural plate, it undergoes depression. This is called neural groove. Undergoes depression, this is called neural groove. At the junction of the ectoderm and neural plate, we have some cells. They are present at the junction of the ectoderm and neural plate. We call these cells as neural crest cells. Neural crest cells. Okay, so what are the neural crest cells? So this is the ectoderm and this is the neural plate, right? And at the junction here, at the junction of the ectoderm and neural plate, some cells, some of the ectodermal cells, they transform into neural crest cells. Neural crest cells. So this is the ectoderm and this is the neural plate, right? So they undergo, ectodermal cells, they undergo transformation, and one different types of cells are formed, that is called neural crest cells. Now what happens here is, so we know that the neural plate, it undergoes depression, and this one we call it as neural groove. Now what happens is the margins of the neural groove, they unite and they undergo further depression and depression. The margins of the neural groove, they come into contact with each other. They come into the margins of the neural groove, they come into contact with each other and forms a neural tube. So neural tube is formed on the top of the notochord by the proliferation. This is the notochord, right? So this is the notochord. So the neural tube is formed on the top of the notochord by the proliferation of the ectodermal cells. So the nervous system is derived from the ectoderm. Okay, so this is the neural tube. On either side of the neural tube, we have neural crest cells are present, which is present at the junction of the ectoderm as well as the neural plate. These neural crest cells, they occupy the dorsolateral aspect of the neural tube. Okay, so what are the derivatives from the neural crest cells? That is very, very important. What are the derivatives from the neural crest cells? Means neurons of dorsal root ganglia, neurons of sympathetic ganglia. These are the derivatives of neural crest cells. Neurons of dorsal root ganglia, neurons of sympathetic ganglia, and neurons of 5th, 7th, 8th, 9th and 10th cranial nerves. Neurons of 5th, 7th, 8th, 9th and 10th cranial nerves, and also they form part of adrenal medulla, and also it's not a 5th, it is a 6th. As you said, 6th, 8th, 9th and 10th cranial nerves, and also it forms of adrenal medulla, part of adrenal medulla, and also chromaffin tissue, it is also derivative of neural crest cells, and melanoblasts, which are responsible for pigmentation, also derivative from the neural crest tissue, and also arachnoid and pia matters of brain, arachnoid and pia matters of brain, and these neural crests, they cranially they undergo into the mesenchyme of the head and neck, and they help in the formation of musculature of head and neck. These neural crests, they also help in the formation of musculature of head and neck. They stimulate the somatomeres of the head and neck, and they help in the formation of musculature of head and neck. So these are the derivatives of the neural crest cells. So the neural crest cells, they form the neurons of the dorsal root ganglion and the neurons of the sympathetic ganglion, neurons of the 5th, 6th, 8th, 9th and 10th cranial nerves, and part of adrenal medulla and chromaffin tissue, and it also forms the melanoblasts, and they form the arachnoid and pia matters. They form the meninges. These are nothing but the meninges of the brain, and also they help in the stimulate the somatomeres, and they help in the formation of musculature of the head and neck. These are the derivatives of the neural crest cells. So how the neural crest cells are formed? So the neural crest cells here, they are nothing but modified cells from the ectoderm. They are formed at the junction of ectoderm and neural plate, and these neural crest cells lie along the dorsal aspect of the neural tube, dorsolateral aspect of the neural tube, neural crest cells. Okay. And now coming to the neural tube. This neural tube, it consists of dilatation part and tubular part, and the dilatation part gives rise to brain, and the tubular part gives rise to spinal cord. At first, this tubular part is very small, and as the embryonic disc grows, this tubular part also grows and gives rise to spinal cord, and the dilatation part gives rise to brain. Okay, so the neural tube, how it is formed? It is nothing but it is formed by the proliferation of the ectodermal cells and forms a thickening called neural plate, and this neural pores undergo depression, and it is called neural neural groove, and it undergoes further depression, and the margins of the neural tube, they combine to each other and forms neural groove, combines to each other and forms a neural tube, which gives rise to brain as well as spinal cord. And we can see that your neural crest cells, and these neural crest cells are very, very important. What are the derivatives of the neural crest cells? Are the neurons of dorsal nucleus, neurons of sympathetic ganglion, neurons of subcranial nerves, and also some part of adrenal medulla, and chromaffin tissue, and melanoblasts, which are the pigment cells, and arachnoid pyometas, which are nothing but the meninges of the brain, and also and helps in the stimulation of the formation of musculature of head and neck. So that's about the neural crest cells and the neural tube. And next, coming to the intraembryonic mesoderm, we know that the intraembryonic mesoderm is developed from the primitive streak. Primitive streak, that is intraembryonic mesoderm, is it goes sideways, and this is the intraembryonic mesoderm. And the center one we call it as, this one we call it as notochord. Above the notochord we have neural tube is formed. This is the neural tube. Now, coming to the subdivisions of the intraembryonic mesoderm. On either side of the notochord, this is the notochord, right? The notochord. This is the notochord, right? So on either side of the notochord, the intraembryonic mesoderm present, we call it as paraxial mesoderm. Paraxial mesoderm. Paraxial mesoderm. Okay. And this mesoderm, we call it as lateral plate mesoderm. Lateral plate mesoderm. And a strap of mesoderm which intervenes, the strap of mesoderm which intervenes between the paraxial mesoderm and lateral plate mesoderm, we call it as intermediate mesoderm. We call it as intermediate mesoderm. We call it as intermediate mesoderm. Okay. Now, so paraxial mesoderm, which is present on either sides of the notochord. On the top of the notochord, neural tube developed by the formation of ectodermal cells, by the proliferation of the ectodermal cells, and this is the neural tube. And here on this is the lateral plate mesoderm. In between the paraxial mesoderm and lateral plate mesoderm, we have a strap-like mesoderm, that is nothing but intermediate mesoderm. We can see here the mesoderms here. This is the paraxial mesoderm, and this is the intermediate mesoderm, and this is the lateral plate mesoderm. Okay. Now what happens here is a coelom appears in the lateral plate mesoderm. A coelom appears in the lateral plate mesoderm, and it divides the intraembryonic mesoderm into two layers, splanchnopluric layer as well as the somatopluric layer of intraembryonic mesoderm. At the top of the procaudal plate, here the coelom doesn't extend. Here the coelom, the intraembryonic coelom doesn't extend. In future, it forms the septum transversum. Which gives rise to diaphragm. Diaphragm, we know that it is a muscle which separates, there is a major muscle of respiration, which separates the thoracic cavities from the abdominal cavities. Okay. So here the intraembryonic coelom doesn't extend on the top of the procaudal plate. We know that this is the procaudal plate, right? And this is the neural tube, and this is the primitive streak. Here we have cloacal membrane is present. On the top of the procaudal plate, the intraembryonic coelom doesn't extend, that is called septum transversum. In future, it gives rise to diaphragm. So, coming to the paraxial mesoderm, we know that paraxial mesoderm is present on either side of the notochord. In future, the notochord it is occupied by on the top of the notochord, neural tube is formed, and this paraxial mesoderm undergoes segmentation. We call this segmentation, we call them as somites. This paraxial mesoderm undergoes segmentation, forming somites. So, out of the somites, 42 to 44 somites are formed. Out of these, some somites at the coccygeal region, they degenerate, and finally 37 to 39 somites are present. At first, 42 to 44 somites are formed. Out of these, some somites in the coccygeal region. We know that this is the coccygeal region. We know that lumbar, this is the thoracic cranial region, and this is the thoracic region, and below that we have the lumbar region, and below that we have coccygeal region. So some somites in the coccygeal region, they degenerate, and finally we have 37 to 39 somites are formed. Each somite is innervated by a cranial nerve. Each somite, it is innervated by a cranial nerve. And coming to the structure of somite, when we take the section of somites, this is the neural tube, right? This is the neural tube. This is the neural tube, and these are the somites. When we take the section of somite, it consists of three parts. The ventral part, we call it as sclerotome. The ventral part, we call it as sclerotome, which gives rise to vertebral column and ribs. And the middle part, we call it as myotome, which gives rise to striated muscles. And the next part, we call it as dermatome, which gives rise to skin, and which gives rise to subcutaneous tissues of the body, which gives rise to dermis and subcutaneous tissues of the body. So this is the somites. When we take the section of stomach, it appears like it. The ventral part, we call it as sclerotome, and the dorsal part, we call it as dermomyotome, which consists of dermatome and myotome. Sclerotome gives rise to vertebral column and ribs, and myotome gives rise to musculature of striated muscles, and dermatome gives rise to dermis as well as the subcutaneous tissue. Okay, and now here the splanchnopodic layer of the mesoderm, somatopodic layer of the mesoderm, and this intraembryonic coelom, it communicates outside with the extraembryonic coelom. This intraembryonic coelom, it communicates outside with the extraembryonic coelom, and the lateral plate mesoderm, it forms all the viscera of the body. The lateral plate mesoderm, it forms all the viscera of the body, and so the entire structures of the human embryo, it is formed by the intraembryonic mesoderm. The intraembryonic mesoderm, it forms the musculature, the soft smooth muscles also formed by the splanchnopodic layer of the intraembryonic mesoderm, and also some connective tissues and sclerotomic tissues, they are also formed by the intraembryonic mesoderm. Subsystems, lymphatic system is entirely formed by the intraembryonic mesoderm. Urogenital system, part of the, except the most of the urinary bladder, prostate and urethra, and sex gonads and sexual organs gonads are formed by the intermediate mesoderm. We know that the intermediate mesoderm is present here in between the paraxial mesoderm and lateral plate mesoderm. Intermediate mesoderm is present, it gives rise to sex glands and nothing but gonads. So all the major tissues of the body, they are developed from the intraembryonic mesoderm. So when we take the embryologies, first we have from head end to tail end, we have septum transversum is there, intraembryonic coelom is there. In future, intraembryonic coelom gives rise to pericardial, pleural and peritoneal cavities. In future, intraembryonic disc gives rise to pericardial, pleural and peritoneal cavities. Here our heart area is present, where heart tube is yet to be formed, and this is the procaudal plate. This is the procaudal, where the ectoderm and endoderm, they are attached with each other, and in future forms the buccopharyngeal membrane, and here neural tube is present. And here we can see the segmentation of the paraxial mesoderm into somites, and here we have primitive streak is present, and primitive streak, below the primitive streak we have cloacal membrane is present. So that's about the formation of germ layers, formation of trilaminar and bilaminar disc, and formation of notochord also we have studied here. Formation of neural tube is also we have studied here, and what is the derivatives of the neural crest we have studied here, and what are the divisions of the intraembryonic mesoderm we have studied here, and what is the somites. So we know that out of 44 somites are present, and these somites correspond to the cranial nerves. We have 8 cranial nerves here, so 8 somites are formed, and we have 12 cranial nerves in the thoracic region, and 12 somites are formed. And one more thing is the somites present in the pre-aortic region, they are called occipital somites, which are innervated by the hypoglossal nerve. These occipital somites, they form the musculature of the tongue. Okay, these somites correspond to the number of spinal nerves, and here the occipital somites, they are innervated by the cranial nerves, and the remaining somites, they are innervated by the spinal nerves. Okay, so these somites, they are innervated by spinal nerves, except the occipital somites, which are innervated by the cranial nerve, that is nothing but the 12th cranial nerve, that is hypoglossal nerve, which are these occipital somites, they give rise to musculature of the tongue. So that's about the somites, and that's about the intraembryonic mesothelium, which in future intraembryonic spilum forms the pericardial, pleural and peritoneal cavities. So that's about the formation of germ layers and fate of notochord, neural tube, neural crest and intraembryonic mesoderm.